Thank you for the opportunity to start with. And as Joe mentioned, I work at the RHS, and I'm a horticultural taxonomist. The RHS has multiple gardens. Our science is in Wisley, and as I work in this shiny new building, which I commandeered one of the largest meeting rooms just for myself. And this is a building for science. It also has libraries and education. As Joe mentioned, the RHS has a rather large interest in education. So the next slide is going to be, again, repetitive because Joe mentioned this. The reason why I'm talking to you, as opposed to any of my colleagues about taxonomy and plants, is that I published the first assembled daffodil plastome in, I think it was 4 years ago now. So this was the poet's daffodil, and John then approached us whether we can expand it, whether he can expand this into sort of school projects, which I guess you are involved in. But that's not the only thing I've done in daffodils. I've been working on daffodils since 2010 with my PhD. I did my PhD on these daffodils. These are called the hoop petticoat daffodils. It's there's a small group of very small daffodils. I did some field work in Spain and Morocco and Portugal, and I'm not going to talk about these right now a lot because I'm going to use them as examples later on. But beyond these, I've also have been supervising two PhD students on daffodils. And so Jordan's work was on this group. It's called the Narcissus minor group. It's really the out of the 4 images, all but the green bordered one belongs to the Narcissus minor group. So the sort of pinkish orange one, the blue and the red. They are rather hard to tell apart. So Jordan's question was his thesis really focused on, can we use molecular tools to delimit these species? And he wasn't as lucky as me, so he never managed to go to Morocco, but he did go to northern Spain and Portugal. And he sequenced a few markers, various different markers, and the results are not very straightforward, as never really in daffodils. And there is a lot of gene flow going on within these plants, so delimiting them is a bit tricky. The major paper, or the biggest paper from his PhD he's still working on it. I'm hoping that it's going to come out next year. Then I have a current PhD student who works on a larger group, as in a more comprehensive phylogeny of daffodils. And as a sort of follow-up from Jordan, where we found that the pattern we see in his results is probably due to repeated glacial periods, so ice ages. So Zoe's work focuses on a large-scale daffodil evolution over time and how past climate change might have influenced that. So for this, she didn't just build, or will be building, a large daffodil phylogeny, but she also did a comprehensive one really on the whole family. So this is the tree. She has one of the most recent ones on the whole family of Amaryllidaceae, where daffodils belong. And daffodils belong in the Eurasian clade, which is on the tree, the second one from the top. And the initial results show that it's about 21 million years old. So I have a second PhD student, a current one, but she doesn't work on daffodils, but obviously I must use the opportunity to mention her work. So Hannah works on hyacinths, squills, and grape hyacinths related plants, these small blue bulbs. Her question is similar to Zoe's. How does past climate change influence their evolution? But also characterizing these in this particular slide, this is her work on muscari, the grape hyacinths. These the 4 photos on the top are roughly the 4 groups we can see, but they are very poorly characterized, so we can't really tell how to delimit these properly. So that's what she's working on at the moment. If you look at the 2 trees on the side, These are color coded, and you might notice that those triangles sometimes split up and move around the tree. So yet again, evolution is within this group either, just as in daffodils, is not really that straightforward. But I'm here to talk to you about how we use DNA in plant taxonomy. But before you do that, and I did mention that I'm a taxonomist, so most of my colleagues and my interest here is to make sure that names in cultivation or plants in cultivation have the appropriate names. We also do some novel research like through the PhD students, but what we are focusing on is naming and classifying mostly cultivated plants. But taxonomy is a bit broader than this, as you could see from the PhD projects. Delimitation, where one group ends and the other starts, is very much part of it. And the idea would be that you delimit and new things and describe and classify them based on some form of shared characteristics. So the most obvious shared characteristic would be, oh, sorry, not there yet. The reason why we classify is, of course, to have communication and to avoid some confusion. But I believe there is an innate human need to pigeonhole things, to create order out of chaos. And to have communication, we obviously name plants. And this is I think the easiest example I could find where communication clearly when you use common names don't work. Both of these plants are called in English bluebell. Both have roughly blue bell-shaped flowers, and depending on how we see blue to purple colors, they are sort of blue. But their Latin name is actually vastly different. One is Hyacintoides, the other one is Campanula, and they are not really even closely related. So their superficial shape that the common names are based on is somewhat misleading. So in taxonomy, we use Latin names like those one on the bottom. And I think this is a good point to apologize. I know very little about the British education system below universities. So if I mention things that you are familiar with, all I can do is apologize. So anyway, we use Latin names in taxonomy to communicate. In plants, the Latin name is always made up of two parts. And these two parts is the genus and the species epithet. You can sort of think about them as a surname, first name combo, where the surname goes at the front. With the caveat that a name of a genus cannot really be shared with another genus. So it's unique, but the species epithet can be shared with multiple species. And this is the Latin name. Taraxacum officinale is the Latin name for dandelion. So the naming and the classification we use originates from Linnaeus in 1753. And it's a very good figure to illustrate the classification that you can imagine as nested bigger and bigger funnels as you move from species towards life. And the bigger funnels contain more and more of the smaller ones. So as I said, taxonomy is partially interested in delimiting organisms, and we would need to use some form of characters. And the obvious one would be morphology, basically what things look like. And my examples at the beginning are rather simplistic to get my hopefully to get my point across. So bear with me. Imagine that you have these two plants. One is orange flowered, the other one is blue flowered, but that's the only difference we can see between them. And if we want to decide how many species there are, it would be sensible to say two. They just look different. But what happens if we find another plant that overlaps in some of the characters, but not all of them? So there are still orange and blue flowers, but there is also a blue one with notched leaf instead of simple ones. So if we go for the same of how many species we can identify here or describe or recognize here, we could say 3, and all combination is validly a species. But what if we say, really, we prioritize the leaves and we don't care about the flower color? Then we would recognize two, or vice versa, we would also recognize two, just a different one. So morphology clearly isn't a foolproof system. It won't always work. And I have some real life examples to show this. And keep in mind for any slide where the title says fails in double quotation or something similar, that really just means in this case, the morphology fails to answer the question we asked in a very obvious manner. So here we have 8 leaves. They are different shapes, different sizes. These are ivy leaves. And if we were to decide based on their look, how many species we could recognize here, we could say the big lobed ones could be one, the smaller ones could be another, and the remaining 2 could be a third species. But in reality, this is really just one species. This is the common ivy, and it's actually from one plant. Ivy has a characteristic where, as the plant matures, the leaf shape changes from the lobed one to the more rounded, heart-shaped one. So just to stay with ivies, I have another example. Here we have 11 leaves, and the question would be the same: how many, based on their look, how many species we could recognize? And obviously there is variation with the leaves, how many lobes and how lobed they are. The sizes in this picture is particularly similar at this stage. We could probably, if we really were pressed for it, circle a few. But really, these are 11 different species of ivies. So just as a gross morphological feature, just the shape and size of the leaf is really not helpful as a morphological character in ivies. So if we can't use morphology, what else can we use? And if we were interested in large mammals, we could use interbreeding as a criteria. So basically, if we have 2 individuals that can produce a fertile offspring, then we consider them belonging to the same species. So the example I have here that where this works is the one I assume you're familiar with, the horse and the donkey. We know that these are 2 different species. They do hybridize. Their hybrid is a mule, but it is infertile, or at least most of the time infertile. Nothing is that simple in biology. So this fits this category. These don't interbreed, therefore we recognize them as 2 separate species. And I was looking for, when I was originally making this talk, I was thinking of an example where interbreeding doesn't really work, which is largely true to most plants. So the example I have is maybe pushing it a bit, what doesn't work really mean, but I think it illustrates some point at least. So here we have 2 larches, the European one and a Japanese one. And obviously they can never really interbreed because the distance between them in the wild is insurmountable for the plants. And they are rightly recognized as 2 species. But if we were very, very strict about interbreeding, if at any one point in time, or really whenever they meet, if they could interbreed and their offspring is fertile, and we were very strict about interbreeding as a criterion, then these should be considered as a single species because their hybrid, Larix martinsii, is in fact fertile. This would be foolish to do. Their geographic distribution kind of prohibits them to interbreed, but they still can. So, and this leads us to the bulk of my talk is, what if we used DNA? Ideally, at least on some level, we could use DNA as an underlying characters that contributes to the morphological variation or their ability to interbreed. And also, as opposed to morphological characters or interbreeding or a host of other options you can use to describe species, DNA provides a lot of characters, and by now, very, very quickly. So the obvious question would be then, how would we use DNA? And at the beginning, at least, and to some extent, this is still a current method, we could use something called DNA fingerprinting, where instead of, we just chop the DNA up into randomly distributed bits. And if those bits that are chopped up on a gel look similar, like in this one, sample number 1, 3, 4, and 5, that have the two bands that are closer to each other, then we could say they are the same or similar to each other. And the other two, where the bands are further from each other, are different. So DNA fingerprinting is not that much used in plants right now. It has been used, and I think it still is, in paternity testing in humans, and also in criminal investigations for DNA analysis. But the shortcoming of this is all we know is the size of the band. We don't really know what's inside that band. So we can't really be 100% sure we are comparing like for like bands. So the next step we could do is we could sequence those bands. And we could basically just look at all the bases that band contains in a sequence, which would be then sequence-based methods. So those of you who are involved in the DaffDNA project won't really see this by now, and I'm very glad you won't. It was rather time-consuming. Before next-generation sequencing, we use the different sequencing method, and we get these electropherograms where you looked at peaks and tried to decide whether it was really an A, or T, a C, or a G. And when we had those, we had a bunch of samples, we created an alignment, what would be on the sequence data on the next to the electropherogram in this picture. And the idea of an alignment is that you compare characters that share the same evolutionary history. In a simpler manner, you can think about it as comparing leaves with leaves and flowers with flowers, as opposed to comparing flowers to leaves. So when you have your alignment and you're happy with it, you can build a tree. So the most common figure probably is a good word here for DNA's use in taxonomy or in systematics. It would be a phylogenetic tree, basically the ones here. And trees come in multiple shapes and various sizes, of course. And these are just some of the shapes we have as trees. The first two, the slanted and the rectangular cladograms, are exactly the same. The relationship they depict is the same. It's just purely a stylistic choice that one is more angular than the other. On the other hand, the third one, the phylogram, contains a bit more information. The length of those black lines is informative. It's proportional to the number of changes within a DNA sequence, for example. So when we talk about trees, we have three important words to use. One of them would be tip, which is effectively just your samples. It's those A, B, C, Ds, and those red dots on the top. And those samples and the tips will be connected to each other through the backlines, which are branches. And then when the branches meet, you have a node that I circled in green. So the node is an important character in a phylogenetic tree because it represents a common ancestor. And a common ancestor is important to evaluate how a tree fits our taxonomic group or any groups. So in this case, the common ancestor of A and B and C and D would be those orange circles, and the common ancestor of all four of them would be that green one. And on this slide, you can notice there is another word called outgroup, which is yet another important terminology really to use in phylogenetics, and I'm going to talk about the outgroup in a few slides. But just to stick to the common ancestor bit, this is a less abstract example with apples, pears, raspberries, and blackberries. And if this would be the tree that depicts the relationship, then the orange ones are the common ancestors of the apple and the pear, and the other orange node would be the common ancestor of raspberry and blackberry, and all four of them are connected to that one common ancestor circled in green. So as a slight detour, the obvious question you could ask is how many samples would I need to have a meaningful phylogeny? Obviously, one sample is nothing, and two samples will always be connected with just a single line. So 3 would be a sensible idea at the beginning, but the only way you can connect 3 samples in a phylogeny would be this one. And this is really the only one. That we won't be able to tell out of these 3 daffodils which ones are more closely related to each other than to the third one. The distance will always be basically one step from the next. So what if we added a fourth sample? And in this case, let's add the fourth sample. That's not part of the group we are interested in. And the group we are interested in is usually referred to as the in-group. So the sample we add that's not part of our in-group is the out-group. So the terminology in phylogenetics at times is exceedingly on the nose. So if we go back to my previous example of having these 3 daffodils, where we can't really tell how they might actually be related to each other, and we added an out-group, this out-group needs to be something that's closely related to daffodils, but not really a daffodil. So in this case, we could use an onion, because the onions are very closely related to daffodils. They are in the same family. And if we build a tree with these 4 samples, this would be the relationship. This relationship, in fact, is correct. I managed to select 3 daffodils that can depict this actual relationship. What the onion basically does is it pulls that daffodil with the white tepals and yellow corona in the middle a bit further away from the other samples. If we were then to use the out group to give a direction to the tree, it's called rooting. If we did that, then this would be the relationship. We can say that the 2 plants closer to each other, the one with the yellow corona, the large trumpet-like corona, and the other one which is more sort of tear-shaped, really, and with the reflex petal, it's called the angel's tears daffodil, are more closely related to each other than the third one. So if I then turn this tree into a rectangular one, then this would be the relationship, and this is a tree I think we are reasonably familiar with. So because I have the opportunity to talk about daffodils, and I've been working on them for over 12 years now, I can never really stop myself not to talk about daffodil diversity and what daffodils really, what they are. So there are about 30 to 80 species. A very characteristic feature is the structure in the middle called the corona. And the corona comes in multiple shapes and sizes, as you can see from these images. Some are small and cup-shaped, some are much more trumpet-like or constricted. There are some that they don't actually have any. The one in the bottom corner, that sort of weird-looking yellow one. There are also different colors. It's usually white from white to yellow, but there is one green species because it can, I think. So daffodils are native to largely the Mediterranean. And they are most diverse in the Iberian Peninsula, so Spain and Portugal. Some of the species, obviously looking at the map, reaches further up north. There is one species that is at least tentatively native in the United Kingdom. And it reaches as far as the Middle East to the east. there are daffodils outside of this distribution, as you can imagine. When you see the daffodils along the road come spring, that happens in other countries. And as you go further east, this is a possibility. There is one described species, or well, it's a subspecies really, in China, but it is not native there. It got there through the Silk Route, and this is a sample you might come across because its plastome has been assembled. But outside of further than the Middle East, the plants you would find are really got there through human influence. So with the RHS and the Daffodil Project in particular, as in the Daffodil Project John started, the interest really is in plants in cultivation. And through crossing these 30 to 80 species over the last, well, sort of 200 or so years, gardeners managed to raise over 30,000 cultivars. That's how many we have in a register that contains all the names. At any one time, there is about 4,000, I think, that's growing in gardens or available in trade. And the concept remains the same. They have the corona, but the corona shape changes vastly. There are some that are split, like the third one on the top, the one that doubles, like the one in the bottom corner. And of course, the colors there is always be limited what's available in the wild. So we really go from whites to yellows, but that one little ring in the poet's daffodil contributed a lot to creating more orange and darker colors, and even that pink one in the middle of the screen. So to get back to phylogenetic trees and daffodils, this is a tree I built during my PhD. And if we were to look for daffodils and their common ancestor, the common ancestor would be that orange circle, and we can conveniently circle the whole group. These sort of circling groups on a tree is a very simple but effective method of looking at how our groups or species or genera, families fit onto a phylogenetic tree. And why this matters, I have a few slides to illustrate, but we need to step away from daffodils because the example I have from one of my supervisors is way too good. Sorry, yet again, I rushed ahead. These common ancestor and circling things, we can go further than just all of the daffodils. We have the green groups here, which are the subgenera. They have their own common ancestors. Of these 6 images, 5 of them belongs to the large group. Only the one in the bottom corner with the white tepals and the yellow corona belongs to the small one. And we can go even further. In daffodils, there is a section. We recognize multiple sections. In this case, this red group, yet again, with its own common ancestor. And that would be the plant with the yellow corona and yellow tepals. So, as I mentioned, circling things is an important important way to check how things fit on a tree. But these are obviously not daffodils. This is snowdrops and snowflakes. They are in fact related to daffodils. They belong to the same Eurasian clade in Amaryllidaceae. And to start with, there were 2 snowdrops, Galanthus, which I assume you're familiar with, and Leucojum, the snowflakes. Fewer plants in that one, also available in gardens. And when they built a tree, I think in 2004, this was their relationship. So you have the two different look of Leucojum on the either side of the tree and snowdrops fitting in the middle. Now, this is not really an ideal situation for naming, because if we were to look for a group to circle all of them and their common ancestor, this would be the answer. The common ancestor would be the one in the orange circle, and it would contain all 3 of them. But if we have a single group, it really needs to have a single name. And in this case, because we have another rule in botany called the priority, So the name that the group takes would be the one that's published earliest. And in this particular case, that would really be leucodium. So all of the snowdrops would need to be called technically snowflakes. This is not really an ideal solution. Snowdrops are much more speciose and more important in gardens. So this is really not a solution anyone would like. So we could look for another common ancestor, and that would be that. Sadly, that doesn't solve it. We can recognize one more genus called Acis, but the snowdrops and leucodium would still fit into the same group with their common ancestor. So would still need to be called leucodium. So the only solution really that is practical really is to split them up into 3. And this is what the authors at the end decided. So now instead of having 2 genus, also 2 genera, leucodium and galanthus, we have 3. We recognize a third one called Acis. So I do have an example that takes us back to daffodils, but it's not as it's a bit less finished because it's a bit more recent, and this is from my PhD. These are on hoop petticoat daffodils. So as I mentioned, hoop petticoat daffodils are small plants, about 15 to 20 centimeters, and they are very recognizable within daffodils. This was one of the reasons I chose to work on them at the time. The size of the corona compared to the flower is much larger than in most daffodils. The whole plant is very small and the flower is only about 1 inch big, but the corona is large compared to the tepals, and it's sort of this cone shaped, which is referred to as hoop petticoat shaped. And we have the usual white to yellow colors. There is the sort of creamy ones on the top. And when I built, and I'm going to bring back the same tree as I had earlier with these 2 groups with hoop petticoat circled on them, but we don't get a single group. We get 2. Sort of like we had leucodium on the tree. The group splits. We have a yellow and a green group. Their common ancestors are circled in orange. And if we wanted to find their common ancestor, the common ancestor of both groups, that would be that blue one. And if we wanted to circle a larger group than these, including the common ancestor, that would be most of the daffodils. So this is not really an ideal solution. But the first question here would be, does these 2 groups fit onto the morphological variation we see, i.e., would it be possible that the 2 groups really just split between the whites and the yellows? That is obviously a question where the creamy one would fit, but instinctively I would say probably closer to the white ones. But when I just circled the plants that went into that tree, this would be the result. So clearly those 2 groups are not really splitting along flower colors. So flower color is not really a useful category, useful character here. Both groups contain whites and yellows. And by now they have been described as 2 different things. One of them is called meridionalis, the other one is bubocodii. And the reason why this probably happens, what we think at this stage, is very complex hybridization through the evolution of daffodils led to this shared characteristic of hoop petticoat shaped flowers. So obviously, because I talked about when morphology fails and when interbreeding fails, I'm bound to mention when DNA doesn't work. Yet again, DNA doesn't work in a way that it just doesn't answer the question we asked in a ready manner. So this is an example from 2006. It's on lupins in the Andes. And I hope you can see the pictures on the side are what those plants actually look like in the wild. Some are small trees, some are small shrubs, some are really just sort of very small plants. And when they build this tree, it's on a few gene regions, if I remember correctly. They can't really resolve that relationship. All you get in that orange group on the bottom is every sample is sort of just connected to a single node. And you can't tell which one of these species are more closely related to each other than to others. The reason behind this, in this case, is with the Andean uplift, as the new ecological niches developed, these plants specialized, diversified very quickly, and took on these vastly diverse forms. And these particular DNA regions that they used in this analysis didn't really have the time to evolve to catch up with the morphological variation. This was obviously 16 years ago, and sequencing technology developed. So we could say that we hope that if we had more data, we get clearer answers. But obviously that's not the case, or not always the case. This is a tree on tomatoes. And what you see on the panel labeled B is hundreds of trees made from hundreds of genes, just purely overlaid on top of each other. And the lines are very fine, but you can see that it's not really a phylogenetic tree, it's more of a phylogenetic shrub. You can, as the authors did, this is a paper from 2016, you can, as the authors did, you can see, as the authors did, highlight a possible tree on top of it as a sort of what has the most support within the data. But still, the underlying picture is not very straightforward. And I have one more example when DNA doesn't really work, when we look at phylogenetic trees, or doesn't really give us a very simple answer. But for that, we need to think of this. In plant cells, DNA contained in 3 different compartments: the nucleus, chloroplast, and the mitochondrion. And the way these are inherited are very well defined, but not really always the same within these compartments. So offspring plants always inherit 1/2 of each of the parents' nucleus, or nuclear genome, really. But the chloroplast genome and the mitochondrial genome is always inherited from a single parent, and it's in plants, usually the female. So the example where to which this is relevant is in daffodils. We have 2 trees here. One is the organellar DNA tree. So it was, I think, 2 chloroplast regions and 2 mitochondrial regions. And the other one is a nuclear DNA tree, which was 1, if I remember correctly, 1 nuclear DNA region. And if you pay attention to those colored triangles, you can see, and the lines connecting them basically depicts how they keep splitting up and moving around across the tree. So since we are working on daffodils and we know they hybridize readily, hence the sort of over 30,000 cultivars, during the evolution of daffodils, there was a huge amount of hybrid activity. So various species crossed with each other, which led to the organellar DNA and the nuclear DNA to track different evolutionary trees, really. So there is interesting questions here, and of course, through Zoe's work with when we expanded the sampling size, well, really the amount of DNA we use, we are still looking into this. This is a paper from Spain in 5 years ago or so. So we'll see how much that might not change. But there is one more thing to keep in mind in DNA, that just because we expect DNA to encode for everything, that's not always the case. So in these particular 3 plants, those characters you see in the flowers, the split colors or the feathering on a tulip and the colors on hydrangeas, are not down to readily sequenceable DNA variation. So the one in the first one with the yellow and red flowers, it's really just small bits of DNA jumping around the genome, switching genes on and off, which creates this mosaic pattern. In tulips, that's just a viral infection that creates the feathering, and the hydrangea flowers going from pink to a sort of paler pink and blues is just a response to the pH of the soil. So if there is one thing I want you to take away from this talk, apart from how lovely daffodils are, is DNA is just one source of data, really. It's on its own to building a tree can answer only help you answer only so far. You always need something else to look at, ideally, but be it morphology or some ecological characters, or even in daffodil cultivars, just any knowledge of their basically family tree. And this is the second time I give this talk that John organized, and this was my last slide last time. Since then, I added a few slides, and I talked to my PhD students of how to summarize their talks, their projects into a single slide, and I asked Zoe about it, and I know she was trying to be funny, and she was funny. Her suggestion was this: daffodil, smiley emoji, daffodil, crying emoji. And she somewhat hit the nail on the head. Working on daffodils can be exceedingly fun, but at times really, really, really aggravating. So this is really the end of my talk. And I think we have about 3 minutes for questions, but I have more time if you have questions, really.
Great, thank you so much, Kálmán. I'll pass this over to Jack and his students. Are there any questions that you've got so far?
Thank you so much for the talk. It was really, really engaging. We've got one question so far, but I'm going to speak for my students if that's okay.
Of course.
I've been nominated. It's when you referred to the term delimiting. It'd be really good. Obviously, as you said, it's about different curriculums, so different levels of language and different competencies around the language of genetics. Would you be able to define delimiting for us?
Delimiting, in that sense, what I meant is basically where you draw the lines. I think the best example I had is where you can circle those things. As in, what would you consider a member of one species versus a member of another species? I.e., what are the boundaries of a species? The other word would be circumscription, but it's much harder to pronounce. So really, delimiting is the best one. What would be a good example? You all work on daffodil cultivars, and let's assume you have, and this is somewhat of a made-up example because daffodils are obviously the cultivars you would work upon on human influence. So you have multiple colors, but the colors are such that they form somewhat of a gradient with some gaps in them. So delimiting a cultivar would be to say everything that's sort of yellow would be one, and that's really delimiting is. It's one where the word, I think, as far as I have only ever used delimiting in the terms in the concept in the context of this, would be just to sort of I drew a circle around it. That's about right. I think that's the best way I can explain it, which is very, very poor, I understand, but I think it feels simpler. I think it sounds more complicated than what it is.
No, I think that's helped. It's just because it was a new term, and I think we've explored it before, but obviously not used that term. So it's quite useful to just throw a net over that one. Thank you. But I don't think we have any other questions. We followed the rest of that. It's really great to give it a wider context as well. Because we've talked about sort of the minutiae of what we'll be doing, but we haven't really talked about how it links more broadly. So it's really great to talk about phylogenetics.
The one thing to keep in mind with your project is that you work on plastomes and with daffodils and with cultivars, but plastomes, the limitation of a plastome is that you can really only track the maternal plant, which is a huge amount of information. It's 50%, and in some cases we don't really know. It's a guesswork, and the samples at the time the cultivars at the time we selected were ones where you could ideally tell which one the maternal parent is, which is a useful information. But that's really it at this stage. We'll see where we end up.
Brilliant. Thank you, and thank you, John, too. It's been really useful.
I've got one more question for you, Kálmán.
Yeah.
So when we think about like mitochondrial DNA in humans, so much of that genome has moved into the nuclear genome. Why hasn't that really happened with plants and in terms of duplications, or as in complete removal?
So it has happened. We don't really use right. So the mitochondrial genome in plants varies a lot in size, much more than plastome. Plastome goes from about 30,000 to about 217, I think the largest so far. Mitochondrium goes from very low tens to 500,000, very large mitochondria. They very readily reorganize, much harder to work with, and exceedingly character poor. So I've built a daffodil tree on mitochondria, not the whole genome, it's very hard to assemble, but most of the genes, it kind of looks like that orange line in the Andean lupins. It has not enough variation. The genes just evolve slower. But so genes did move from mitochondrium from the nuclear, but it's much more documented in plants on plastomes. So if you look at algae, the plastomes of algae, they are about twice the size of land plant plastomes, and the genes didn't get lost, they just got transferred to the nucleus. So you start from 300 and something thousand base pairs, which is now down to about 160. So quite a lot of the genes moved. The why I won't be able to answer. It's much more, if I remember correctly, if I understand correctly, it's much more efficient to maintain the nuclear genome than multiple copies of the chloroplast. You have thousands in one chloroplast, and then thousands and lots of chloroplasts in a cell, then lots of cells. So it's much easier to maintain nuclear genome, but it is much bigger. So that's really, I think, it.
And you mentioned one thing about the problems of assembly. One of the Genetic Society was about the idea of a branched plastome sequence and linear forms as well as the circular form.
Yeah, so the idea was at the beginning that the plastome is a circular molecule. And I think I think less than 10 years ago, someone showed that this is actually it's not entirely correct. It does exist as circles, it does exist as linear versions, and it also exists, which my favorite one is linear concatenators. The only time I've seen this, basically you have linear molecules that just keep stitching one after the other, like basically train cars of plastomes. Why this matters is how the molecules can recombine and form newer and newer variants. But for simplicity, especially during your project, and really for no other reason, if nothing else, than saving space, a circular plastome is much more efficient to illustrate a whole plastome than a linear one. It's just easier to see. Plus, you can see the inverted repeats and various other things in much clearer. But yeah, they do exist, and then people in papers keep fighting each other about it. I'm not sure why, but they do. It does matter how they recombine, which is rare still.
Great. That's all of the questions I have. Jack, unless you've got something else, I think it's just a big thank you to Kálmán. Thank you so much for giving up your time today.
Thank you very much for the opportunity.
Yeah, thank you.
And we'll let you get on and enjoy your evening.
Thank you.
Thank you, everyone.
Bye.